The overall goal of this procedure is to site-specifically introduce chemical probes into an antibody fragment by genetically incorporating an azide-containing amino acid.
This method can help answer key questions in the chemical biology field, such as in protein modification, engineering, and conjugation.
The main advantage of this technique is that your target probe or drugs can be site-specifically introduced into proteins or antibodies by simple mixing.
After constructing the plasmids, add 1 microliter of each to 20 microliters of E. coli strain DH10 beta at a concentration of 2 × 10⁸ cells per microliter to a 1.5 milliliter centrifuge tube. Use a pipette to gently mix the solution. Then place 20 microliters of the mixed solution into a clean cuvette. Set the electroporation machine to 25 microfarads and 2.5 kilovolts. Then, insert the cuvette into the slide of the shocking chamber. Push the slide into the chamber until the cuvette makes firm contact with the chamber electrodes. Press the pulse button once until the electroporation machine beeps. After this, add 1 milliliter SOC medium to the cuvette quickly. Then, transfer the mixture to a test tube. Incubate with shaking at 37 degrees Celsius for 1 hour. After incubation is complete, spread the transformed E. coli cells onto lysogeny broth agar plates containing a